I'll just say welcome, everyone. My name is Susan Law. I'm the Long COVID Web Knowledge Mobilization Lead, and I'm delighted to welcome everyone to today's webinar. It's part of a monthly series that we offer through Long COVID Web. And I will just also introduce Dr. Angela Chung, who's here as the nominated principal applicant for the network. So welcome, Angela, and to our guest speakers today.
Thank you.
Just say a few little introductory comments here, and then we'll get started. So if we'll start with a land acknowledgement, we wish to acknowledge this land on which the Long COVID Web operates. For thousands of years, it has been the traditional land of on the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. Thank you. Just a quick recap of the purpose of the Long COVID Web webinars. It's to share evidence-based information, research results that are relevant to decision makers, researchers, clinicians, patients and families, and the general public, to provide ongoing updates, spotlights on emerging evidence and scientific information about the activities of our national network, and to contribute to reducing and managing misinformation about long COVID. We do have a code of conduct. The link for that is at the bottom of this page. And we do very much highly value engagement around the science and its interpretation, as well as its application. And a commitment to reasoned discourse, as stated in our code of conduct, comes with the responsibility for all of us to ensure that any comments or questions remain within the spirit of positive, constructive, and respectful discussion. We welcome any or all questions and comments in this spirit. Thank you. So you note that the cameras and mics are off, so please post your questions in the chat. We will pause for a question and answer session after each speaker, and we will do our best to answer as many questions as possible. These sessions are recorded, so your attendance assumes consent to recording, and with permission from the speakers, the slides may be posted online following the webinar. In this case, because we have two excellent graduate students who are working on their research for their theses, we will not likely be sharing these slides, but we can confirm what can be posted afterwards. And we do welcome your thoughts about how these sessions could be most helpful to you. We've designed a very short feedback survey, which will be posted during the seminar towards the end. So thank you. If you'd like to join a Long COVID Web and you're not a member, you can click on this link here and join so that you receive notices of these meetings. So I'm going to end there and turn it over to our speakers, Estefanía, Espin, and Elizabeth Guinto, who are both graduate students at the Heart-Lung Innovation Center at the University of British Columbia.
Good morning, everybody, and thank you, Susan, and thank you, Angela, and to all the people who make the Long COVID Web for giving us the opportunity to present our studies here. We are very glad to have this chance. I'm a doctoral student in the experimental medicine program of UBC, as Susan mentioned. And we, in the group where I'm working, we have, well, my supervisor and the group I'm working with has experience in developing biomarkers for different diseases. So my thesis project is aiming to identify biomarkers for long COVID, but also to better understand the pathobiology of this disease. So there's a study that I'm about to present is a long COVID in paramedics and exploratory gene expression analysis. It's a pilot study, and we will for sure advance on these findings. I would like to start with the land acknowledgement. I acknowledge with gratitude that I live and study on the traditional, ancestral, and unceded territory of the Musqueam, Squamish, and Tsleil-Waututh nations. About long COVID, as all of you might be aware, this is a debilitating illness that occurs in at least 10% of the COVID infection survivors, which accounts for up to 65 million individuals worldwide, and this condition manifests across different organs and systems, and those symptoms can last from months to years, and some of them are even expected to be lifelong, as the chronic fatigue syndrome. The risk factors are an older age, female sex, a higher BMI, presence of comorbidities, socioeconomic deprivation, and also belonging to ethnic minorities. There are no current treatments and also no diagnostic tests. Actually, the diagnosis is made by the exclusion of other conditions. Therefore, identifying long COVID biomarkers could be crucial for improving diagnosis, but also understanding the pathobiology and probably defining treatment responses, considering that it is a very heterogeneous condition, and there might be different entities of long COVID even at the molecular level. So there are multiple potentially overlapping causes of long COVID, immune dysregulation that could be due to the viral persistence, and also reactivation of other viruses, microbiota dysbiosis, autoimmunity and immune priming, blood clotting and endothelial abnormalities, and also dysfunctional neurological signaling. Then this CORSIP study was a study across Canada, aiming to assess the occupational risks and the seroprevalence and immunity of COVID-19 among paramedics. The researchers in British Columbia contacted us to have the opportunity to collaborate with analyzing PAX gene samples that they could collect from their study. So their study started the recruitment in January 2021, and the participants were followed up with questionnaires each 6 months, and the study ended in January 2023, where they could gather these blood samples for us to analyze. Then the long COVID cases definitions for this cohort where the cases were identified as long COVID, those who met both criteria, having new symptoms lasting 2 months or longer that they did not have before 2020, and also these symptoms continued from a COVID infection or started within 3 months of a COVID infection, which is the WHO definition of long COVID. Then from the samples that we were provided, we identified 8 long COVID cases and selected 33 controls. All of them were COVID positive, which means that the controls are recovered individuals who did not develop sequela. And then we conducted the gene expression analysis using the pan-cancer immune profiling panel of NanoString Technologies. Regarding the characteristics, these long COVID cases had a median follow-up of 10 months. The symptoms duration varied from 2 to 34 months. Not all of them were vaccinated before the COVID infection, and the symptom severity varied from mild to moderate and severe in some of the cases. Then we balanced for all the demographic and clinical variables between the case and control groups, except for age, which was really hard to balance. However, in the logistic regression, only in the univariate analysis, it didn't seem to mean a higher risk of developing long COVID just by the age. Then the comorbidities were also balanced between the both groups. And then what we had in the end is we assessed the genes, the differentially expressed genes. And here I'm presenting a volcano plot where you can see the genes that are differentially expressed in long COVID cases compared to the control group. So in this right side, we have the genes that are upregulated in long COVID cases, And in the left side, we have the genes that are down-regulated in long COVID cases compared to the control group. We identified a total of 73 genes that were different between long COVID cases and the control group. And then we identified if these genes were part of signaling pathways that would be relevant for the pathobiology of long COVID. And we found some interesting pathways here. We have antigen processing and presentation. We have the neutrophil extracellular trap formation. These were down-regulated, platelet activation, focal adhesion, viral myocarditis, phagosome. These in KEC database and in Reactome database, we found similar pathways that were also dysregulated. Platelet degranulation, neutrophil degranulation, platelet activation. So most all of them related to probably viral persistence due to antigen processing and presentation, but also to probably endothelial abnormalities and thrombosis. Therefore, I highlight some of the genes that seem to be interesting and also make sense with the pathobiology. And it's interesting because it has been reported that SARS-CoV-2 proteins are capable of directly interacting with the host proteins, down-regulating them for their viral replication benefit. And we identified in our study that the mitochondrial antiviral protein is down-regulated, which allows the viral to reduce the subsequent expression of NF-κB pathway and also the production of the interferon gamma. And likewise, there is upregulation of phagocytosis and autophagy, which are genes that also are upregulated, and this would allow the virus to have this media, the phagolysosome, for promoting the viral replication. We have down-regulated this gene that is related to the lysosome activity, and it is also a marker of degranulation in NK cells, in natural killer cells. This is upregulated, and it seems to be one of the receptors that facilitates the entrance of SARS-CoV-2 in the host cell. Similarly, regarding the immune cells, it has been reported that lymphocytes are in an exhausted state, possibly due to viral persistence or chronic infection, or to the continuous presentation of antigens. So we had immune checkpoints as BTLA, which is a marker of exhaustion that was upregulated. Also CD47, that is antiphagocytic, which allows, this marker is also expressed in other cells, allowing to evade immune response to infected cells. And some other molecules, as CD74, which is an activator of central memory effector, cytotoxicity of lymphocytes and proliferation was then regulated. But also we had some other CD molecules, like CD2, that were upregulated, meaning that there's activation of the CD cells, but also exhaustion. This is a nuclear factor that is related to the differentiation of lymphocytes that was downregulated, and this leads to exhaustion of these cells. Interestingly, also apoptosis pathway seems to be downregulated in these cells. TP53 was downregulated, which is a pro-apoptosis protein, but also other proteins like ATF2, which is anti-apoptosis, is upregulated. So it seems that the virus is influencing probably the activation and the downregulation of some pathways. There is also TGFB1, which is related to fibrosis, which is downregulated, but this protein is also related to the microvasculature integrity. And being this downregulated could be also a signal of the endothelial abnormality that is present in long COVID individuals. We also have this molecule that is upregulated, which is a cytokine mediator of inflammation, and has also been reported to be upregulated in individuals with long COVID and obesity. So it seems to create a proper media for inflammation and the viral replication and persistence. And also the colony stimulator factor is downregulated. And regarding clotting and endothelial dysfunction, we can see here that thrombospondin, which is directly related to endothelial integrity, but also its downregulation could be related to ROS, to the generation of reactive oxygen species. And also we have downregulated P-selectin. Both of them are also part of the granules of platelets. And on the other hand, we have these other inflammatory markers that are upregulated, specifically this that is related to the neutrophil chemotaxis and adhesion, which could be related to the neutrophil extracellular trap. And then we have some other molecules that are related to the complement system, but also to the platelet and endothelial integrity that are mainly downregulated in these patients. So regarding this pilot analysis, we identified at least 73 genes that were differentially expressed in long COVID individuals compared to controls, but it's also interesting that these genes do not seem to be just randomly differentially expressed, but they seem to be part of pathways that are relevant for the pathobiology that has been proposed for long COVID, as autophagy, phagosome, antigen processing and presentation, focal adhesion, platelet activation, neutrophil extracellular trap formation pathways. So these might be potential candidate biomarkers of long COVID. So our objective now is to validate this in bigger cohorts, and also considering the importance of this cohort, because this is a young population, non-hospitalized group of paramedics. We thought that it also would be interesting to have a longitudinal analysis of these individuals, and we aim to evaluate the level of these biomarkers prior the infection and then during long COVID to compare if there's like an actual response after the infection that could explain all these alterations. So I would like to acknowledge my supervisor, Dr. Scott Tebor, my supervisory committee members, the CORSIP study collaborators, and also the members of the TEBOTS lab. I'm grateful and thank you for your attention. I'm happy to answer any question.
Thanks very much, Estefanía. Estefanía, that's terrific. And an excellent overview of a very complex situation and set of factors in looking at biomarkers. Do we have any questions for Estefanía? Well done. Yeah.
I don't see. Oh.
There we go. From Lorraine. If viral persistence is still one of the hypotheses for a post-COVID condition, why have neither of the trials of antiviral antibodies, Paxlovid, been found to have no effect?
Yeah, it seems that probably the answer to that could be that there might be different phenotypes of long COVID and probably different endotypes, meaning that the cause of long COVID in some individuals could be viral persistence, but not in all of them. Actually, in the studies where they have measured viral persistence, they have found it in half of the individuals with long COVID, in which individuals viral persistence could be one of the factors and the causes of long COVID, but viral persistence has not been found in all individuals with long COVID, and it has also been found in individuals without long COVID. So it might be a cause for some of the individuals, but not for all of them. And probably that's one of the reasons that I think that therapies should be targeted, and that's why biomarkers should be relevant, because it's not just one condition. We have different presentations and probably different causes. So it's my speculation that probably why Paxlovid trials have not been demonstrating results in all the individuals is probably because of that. Subclassifying individuals could be better for addressing their efficacy.
Very good. Thank you. So from another participant, so would the upregulation in inflammatory markers on a cellular level cause the RF to increase in blood work?
I would like to know what RF is the abbreviation for.
I think it's rheumatoid factor.
Oh, okay. Well, it might be also the neutrophil, the NETosis has created also an environment where autoantibodies and autoimmunity could appear. I have not done much research on rheumatoid factor, so could not explain that, but I think that it creates a media for autoimmunity that could be related to that.
Thank you. And there's I'm not sure if it's a comment or question. Antiviral tissue penetration is poor or non-existent. Is that in response to the other question?
There are different studies that have been published. Some of them have been done in blood, but the main findings of viral persistence actually has been in tissue samples. It seems that it penetrates tissues. Probably endothelial tissue will be the most affected, but also it has been found in different organs, even in brains, in autopsies of individuals who had COVID and then some of them sequela. So it seems to be a viral infection. We don't know if it's actually the whole viral or viral particles. It seems that they have found subgenomic RNA, which is an indicator of viral replication. But actually in our pilot study, we included SARS-CoV-2 genes, aiming to find them, probably upregulated or trying to find them. We couldn't detect those genes. It might be that there is no viral persistence, but also it could be a limitation of the technology we use, because it would be better with PCR, quantitative PCR. However, we tried also to detect that. It might be harder to find them in circulating blood. And for Patricia, I will for sure. I will try to give like a version that could be shareable of the slides to the long COVID web. But for sure, I will publish and happy to share it with anyone who needs it.
We can help with that when it's published. Our newsletter
that would be amazing.
Yeah, that's
Thanks.
Great. Good. Patricia gives you a thumbs up there. That's great. Another comment, what is the most significant change in biomarkers after vaccination in long COVID patients?
Well, my study was not designed to answer that question. We had some individuals vaccinated, some of them were not. It was not planned for assessing that. What I only can say about vaccination is that from systematic reviews and studies, it seems that vaccination reduces the risk of long COVID, but which biomarkers have changed due to vaccination is not, I have not evaluated that.
Thank you. So maybe we could just end before we turn over to Elizabeth, but just tell us a little bit about how your work then fits in with the larger program of research at your center. What do you hope will happen to these results?
Well, I have some different paths that I'm going to follow. The idea is that we also will be collaborating with Elizabeth group. They have RNA-seq results of probably a bigger cohort of respiratory long COVID individuals. That will be a second cohort for validating if actually in those individuals also these biomarkers are dysregulated. The other path is evaluating these biomarkers at the protein level, which will be a confirmation that these are actually dysregulated in long COVID patients. And if those findings are positive, then we could move probably to validate them in a bigger cohort. But also we're interested in the dynamics of the biomarkers across time. We think that that's one of the advantages of the CORSIP cohort, that they were able to gather individuals at the beginning of the pandemic, and they are able to have different samples, and that allow us to have like a basal level of molecules, which is interesting for better understanding the pathobiology. And I also would like to mention a brief promotion. I have considered also that it's interesting and is primordial to have the patient's voice and the clinicians who are diagnosing these patients' opinions and expertise in the biomarker research. So I'm about to start a qualitative research also to interview patients and interview clinicians involved in the diagnosis of long COVID, because we consider that they would have a better idea if the phenotypes are interesting, if the endotypes are interesting, and what would they actually expect from biomarkers. So that's about to start. I have also approval, so I will be also happy to share with the long COVID web if they would like to participate in that too.
That's terrific. Music to my ears, because I'm a qualitative researcher. So good luck with it.
Awesome.
All the best. So that seems to, we might have time for these last two quick questions here, although they might be longer than we have time for. But the first is, could this lead to a blood test for long COVID?
Could this lead to, yeah, that would be like the ideal future of this research would be to be able to have a test and also to be like a blood test that will be easy to implement for diagnosing long COVID patients. So yeah, that would be our ideal. It might be a long way until we get that, but yeah. The other is mentioning about the receptors, toll-like receptors. Well, I have not focused mainly on those. I was more interested probably in the thrombosponding pathways and these that seem to be endothelial damage, although it might be relevant because we have the mitochondrial antiviral signaling protein downregulated, which could be related to further activation or prior activation of these receptors. It might also be relevant for the disease. And what I think that might be interesting is that also some of these pathways, or most of these pathways, have molecules or inhibitors that are being studied that have a pharmaceutic presentation, and that could probably aid in the treatment of patients if we have a clearer idea of the mechanisms of the pathobiology.
Great. Well, thank you very much, Estefanía. That was terrific and well done. And thank you so much. We'll turn now to Elizabeth and her work on immunophenotyping. Thank you. Thanks everyone for your questions.
Thank you everyone for attending today. And I'd also like to give a special thanks to Estefania and Dr. Tebbit for recommending me for the second presentation, as well as Susan and the Long COVID Web for making this all possible. I'd also like to acknowledge that I have the privilege to live, work, and play on the territories of the Musqueam, Salish, and Tsleil-Waututh nations. So I'm Elizabeth. My supervisor is Dr. Don Sin at the Center of Heart-Lung Innovation, which is at St. Paul's Hospital, a campus of the University of British Columbia. And today I'll be sharing some of our single-cell immunophenotyping data we have on pulmonary long COVID patients. And so I don't think long COVID needs an introduction to this group. But one thing I'd really like to highlight is that it's such a heterogeneous disease. It affects multiple organ systems, different individuals in different ways with different severities. And so this large review published by Subramanian et al. Found that when they did clustering of these symptoms, that the specific class 2 kept popping up, which was primarily dominated by respiratory symptoms such as cough, shortness of breath, and phlegm. And we are a respirology lab. So we decided to use that to try and subtype this phenotype of long COVID into pulmonary long COVID. And so we've defined pulmonary long COVID as new or worsening pulmonary symptoms after COVID, 3 months, with the duration of at least 2 months after the disease. And so this might consist of dyspnea, cough, chest pain, phlegm, and other respiratory symptoms. The other definition we've put is that they must have a St. George's Respiratory Questionnaire, or SGRQ, score of greater than 10. The SGRQ is a validated questionnaire typically used for obstructive pulmonary diseases, and essentially a score greater than 10 indicates that these symptoms are really impacting these patients' lives. It's a relatively long but pretty simple questionnaire. And so we've hypothesized that pulmonary long COVID is caused by an inflammatory response, specifically in the small airways. Whether this is due to residual virus or autoimmunity remains to be seen, but we are looking at avenues to investigate these. So in order to answer this hypothesis, we developed the response cohort. This is a cohort of about 100 individuals with and without residual symptoms after COVID that is community recruited through social media, as well as through our post-COVID clinic at St. Paul's Hospital. We then administered a variety of questionnaires, including the ones on the screen, such as the SGRQ, the Euroqual, the Pittsburgh Sleep Index, but we've also administered some post-COVID questionnaires, as well as the COPD assessment test and other respiratory questionnaires. We then collected blood from these patients, both serum and peripheral blood mononuclear cells, as well as pulmonary function testing and imaging, lung imaging, specifically xenon MRI, as well as computed tomography. And finally, a subset of eligible and consenting patients underwent bronchoscopy, where we were able to collect bronchial brushings, which contain airway epithelial cells, as well as bronchoalveolar lavage, which contains airway immune cells. And so I'll start by going into the blood, where we did some proteomic analysis using mass cytometry. The cells that we profiled in the blood were specifically peripheral blood mononuclear cells. So these cells are immune cells, so they don't include red blood cells or platelets. And they are also mononuclear, so they exclude your neutrophils, eosinophils, basophils, and other granulocytes. But they do contain lymphocytes and other myeloid cells like monocytes and dendritic cells. So it's a specialized subset of immune cells that we are looking at. And we are quantifying these using mass cytometry, which is also known as cytometry by time of flight, or CyTOF. And to keep it kind of short and sweet, the cells are basically incubated with antibodies against markers of interest on the cell. And those antibodies are conjugated or bound to metal isotopes. And what we do with these cells that are bound to these antibodies is we zap them with this laser that essentially vaporizes the cells, and those elements, those metal isotopes that are conjugated to those antibodies, can then be analyzed through this detector. And so based on their mass and their charge, we're able to see which cells are positive for which antibodies. And that in turn tells us what was on the cell surface and what protein markers were there. And this lets us determine cell type, but also different activation markers. So it's a very valuable tool. And to analyze this data, because it's quite complicated data, we have up to 38 markers, millions of cells for a bunch of different patients, and they all have different disease statuses. So the overall process involves a pre-processing or quality control step to remove kind of unwanted elements. Then we transform and kind of normalize the data. And then we add metadata to define, what are the markers of interest we want to look at, remove the empty channels, and add disease status and patient information. We then cluster the data. So cells that look similar are one cluster. So we're able to kind of see the different cell types. Then dimensionality reduction is performed, and that makes the data more readable to the human eye, essentially. And then finally, differential analysis lets us look at the differences between patients of different disease states. So this is our cohort for CyTOF. We have 30 patients, 17 of which met our definition of pulmonary long COVID, and 13 of them which are control patients. Of the control, 12 of them had mild COVID, and one of them was unexposed to COVID at the time of their blood draw. And as we can see, they are relatively similar, other than COVID-19 severity and SGRQ symptoms, or SGRQ scores. And as we can see, again, with SGRQ, we're using that to define pulmonary long COVID, so it's expected to have some difference. And then in terms of COVID-19 severity, hospitalized individuals were more likely to be in our pulmonary long COVID cohort. And so when we actually look at the data, this here is a UMAP. So the way to read it is every dot on this plot is a cell, and depending on the color of that cell, they are different cell types. So for example, that red cluster at the top, that's a B cell. And so this here is kind of the big generic immune cell clusters with kind of the bigger cell types. And in order to save computational space, this UMAP only represents 30,000 cells, so 10,000 cells per patient. But we were able to do the analysis on all 8 million cells we were able to obtain after quality control. And so here is a stacked bar chart with more of the cell subtypes where we really tried to define the different cells. So for example, for CD4 cells, we were able to differentiate effector versus memory. And this is done on all 8 million cells to look at the different proportions. And what we found was that was particularly interesting is that there were 3 cell types that were significantly increased in our pulmonary long COVID group. And these were double positive and double negative T cells. So these are T cells with both CD8 and CD4 expression. They're also T cell precursors. They're kind of baby T cells that will eventually grow to be mature T cells. And also myeloid stem cells, which again is a precursor cell, were increased. However, in the blood, at least, we were not able to detect any T cell, a mature T cell or a myeloid cell subsets that were increased. This to me indicates that these stem cells are probably entering the tissue and then differentiating, or they're differentiating into something that our CyTOF was not able to capture. And so with that, I'd like to lead into the single cell transcriptomic data we have of the airway immune cells. So single cell RNA sequencing is a technology that lets us look at all of the RNA in an individual cell and compare it to the neighbor cells. And this is a really good tool to look at cell populations. And we specifically use the 10x Genomics platform, which essentially has the cells running through a microfluidic system where the cells will each be captured in an oil droplet with barcoded gel beads that contain all the reagents necessary for reverse transcriptase PCR. And so once we have all those gel beads in the oil mixture attached to a cell, we put all the necessary conditions for reverse transcriptase to occur, and then we get all these transcripts. And so each cell that was with a droplet, all that RNA will turn into cDNA, and all that cDNA will be barcoded depending on which droplet was with that cell. So that when we remove all the oil and we pool all that cDNA together, we're able to track back which cDNA belonged to which cell based on that barcode. And we did this on bronchoalveolar lavage cells. So for bronchoalveolar lavage, or BAL, the patient is put under conscious sedation for bronchoscopy, where a camera is essentially put into the lungs. When we want to get the BAL, we use either CT-guided imagery to determine where we want to get the BAL, or it is at the bronchoscopist's discretion. The bronchoscope is wedged in the area of interest, and then saline is instilled into the lungs. There's a dwell period given to give the time for the saline to kind of stay for a bit, and then it is collected in a trap, and it contains all the immune cells that are kind of circulating around the lung. And then for processing, once we have that, is a bunch of filtering steps, and essentially washing steps. And the point is to be as least stressful to the cells as possible. And then we submit up to 10,000 cells for single cell sequencing. When we do the analysis, it's actually very similar to CyTOF. We have this big, large, multi-dimensional data set. So we do SOUP-X to correct for ambient RNA. So that's RNA from dead cells that had died before our analysis or in between the bronchoscopy and our processing. We do, again, quality control steps to remove cells that we're not interested in, normalize and batch correction, doublet identification, and then clustering, and then cell type annotation. And so our bronchoscopy cohort is, of course, a little smaller because not all patients are eligible or consenting. And so we were able to get 7 pulmonary long COVID patients and 10 controls. And again, relatively similar, other than the SGRQ score, which again is what we're differentiating on. And with the bronchoscopy samples, we're a bit more balanced in terms of COVID-19 severity. So here's another UMAP. So again, every dot is a cell. We were able to get 139,000 cells total after quality control. 44,000 of those belonged to the pulmonary long COVID group. And so if we look, it is primarily dominated by alveolar macrophages, notably M2s, which are considered the homeostatic anti-inflammatory phenotype. We also were able to get T cell and dendritic cell subsets, as well as B cells. We did have mild epithelial cell contamination. However, this is expected, though for downstream analysis, these epithelial cells were removed from our data. And so again, when we look at cell proportions with single cell, because we're looking at a smaller sample size, we actually use the SC-CODA Bayesian algorithm, which instead of giving us a significant p-value, tells us if the differences in cells is credible or not. And so in our case, pulmonary long COVID had a credible increase in CD4 T cells. And when I subsetted our pulmonary long COVID group based on SGRQ score by tertiles, where the third tertile had the highest SGRQ scores, being the most symptomatic, the most sick patients, they had the higher proportion of CD4 T cells, whereas T1s, which were the least sick, were closer to controls. Unfortunately, again, due to small sample size, we're not able to do statistics, either Bayesian or frequentist. But I think here the difference is quite visually different. So it seems like the T cells have something to do with this. And so T cells are a part of our adaptive immune system. So they can, develop memory. And in order to work, they need to have antigen that is presented to them. So they take, pieces of virus, other cells will take pieces of virus and present them to the T cells, who will then look at that piece of virus through MHC and then activate various signals. So when we looked at all the T cells in the single cell of the BAL, we found that they were highly activated in pulmonary long COVID with increases in interferon signaling, adaptive immunity, and TCR signaling. In the CD4 T cells specifically, there was a lot of interaction pathways, cell surface interactions, immunoregulatory interactions, as well as, again, that interferon signaling. For CD8 T cells, we had responses to stress and stimuli, as well as apoptosis and other, again, high immune pathways. So we looked at those antigen presenting cells, and the premier poster child for antigen presentation is dendritic cells. And they had only one pathway upregulated, which was interferon signaling. So we decided to also look at other antigen presenting cells, so the alveolar macrophages in particular, and found that they had an increase in the pulmonary group in cytokine signaling, adaptive immunity, and toll-like receptors. I saw that question earlier. So they're clearly recognizing something that is leading them to be presenting all of these signals that are related to inflammation. So I decided to use the cell phone algorithm to look at the interactions between cells. In pulmonary long COVID, there were up to 12 significant interactions per cell pair, and they tended to be in the more pro-inflammatory phenotype cells, so CD8 T cells, monocyte-derived macrophages, the CCL3 positive alveolar macrophages, gamma delta T cells, these types of cells, and they were very talkative with each other. But when we look at the controls, they're a lot less talkative. There's only one significant interaction per cell pair, and they tended to be on the homeostatic side. So on the metallothionein positive alveolar max, the M2 alveolar max, and the proliferating alveolar macrophages. So in conclusion, we found that in the periphery, we saw an increase of T cell precursors and myeloid precursors, indicative of neutrophilic and cellular immunity, which in the airways was confirmed. So a colleague of mine recently published recently submitted revisions on her manuscript detailing the single cell brushings that we got. And she found that there was increased neutrophil infiltration in the small airways. And in the BAL, I'm seeing increased CD4 T cells in the airways that correlates with symptoms. So these myeloid and T cell precursors that I'm seeing in the blood seem to be differentiating in the airways into neutrophils and T cells. Our next steps are to do CyTOF on the BAL to look at the protein data that we got from our single cell RNA-seq findings. And then we are sending some samples to Texas to do an autoimmunity panel in serum to determine if this inflammation is due to autoimmunity. And we're also looking at bioinformatic tools for retroviral detection, such as herpes simplex virus or Epstein-Barr virus. We did look at both our bulk sequencing and our single cell sequencing raw files to see if we could find any COVID-19 mRNA, and we were unable to find it in the respiratory tissue. So we're looking to see if maybe it is actually a different virus that got activated in the wake of COVID-19. And I'd like to end by, again, acknowledging everyone on the slide, my lab, Dr. Janice Leung, our bronchoscopist, the McNagny lab for their help on CyTOF and helping with the immunology findings, the Biomedical Research Corps for all their help. And I have my email on the screen. If I'm unable to get to your question today, please feel free to shoot an email. Same thing if you're interested in collaborating or talking more about the data, I'm happy to share. Thank you very much.
Terrific work. Thank you so much, Elizabeth. Well done, and to both of you, very well presented. Thank you. So we have a question from Lorraine, and others feel free to post any questions you have in the chat. Since you are seeing increased CD4 T cells, have you looked at post-COVID condition in post-solid organ transplant patients and others who are on meds to deplete their CD4 T cells? What is the prevalence PCC in that population? Is it different? I think perhaps it's prevalence, yeah.
Thank you, Lorraine. That's a great question. Actually, we unfortunately don't have that cohort, so I wouldn't be able to tell, but I can definitely scour the literature and see if organ transplantation does affect COVID-19 and pulmonary long COVID in particular. The CD4 T cell increase as well was in BAL, which I don't know if getting BAL from these transplant patients is necessarily possible or wise, but I'd definitely be interesting to see if that data is available in the literature. It wouldn't be something we'd be able to do here, unfortunately, due to ethics, but definitely something to look into. Thank you.
Thanks. So maybe I'll ask the same question I asked of Estefanía. Just in terms of the bigger sort of lab work that you're involved in your piece, what are you hoping will happen with your results and in terms of contributing to that bigger research program and results?
Yeah, and that's a fantastic question. So my supervisor, Dr. Sin, is actually a respirologist who does see lots of patients, including everyone from the response cohort did get to speak to him. And so I do believe that with his help, as well as other respirologists, I've spoken at a couple conferences where I've got to meet some physicians, and I'm hoping to give them something that they can tell their patients what's happening. I think that's a big thing with long COVID is physicians can try and explain things to their patients, but it's very difficult, especially when, and I didn't show this today, but all their pulmonary function tests, a lot of times their CT scans come up completely normal. And so these patients don't feel hurt or don't feel like there's a mechanism behind it. So even if I can just share, like, this is what we found, for that physician to offer some reassurance to the patient so it doesn't feel like a bunch of question marks. I'd really like to see that more and more.
Yeah, that's terrific. And actually the images are very telling as well. So it's a good way of communicating. So we have another question. Are there differences in CD4 plus T cell subset populations between the control group and long COVID?
Yeah, thank you for that question. So for the CyTOF, I have not been able to find them, but I am still working on really trying to get very granular with those cell groups. In the long COVID and the single cell data, unfortunately, we don't have enough CD4 T cells that I think we can really subset them. That said, I currently have a colleague from the McNagny lab that is looking to try and see if he can differentiate, Th1, Th2, and Th17. And he seems to think that there might be a Th17 signal that we might be able to see. So hopefully to come soon.
Great, thanks. Another question, some comments from Patricia. Are there any plans for clinical doctors to be exposed to this research? As a long COVID patient, I'm very concerned how patients are being directed to only meditation and being told that long COVID is an anxiety issue that can be treated with neuroplasticity, mindfulness practices, instead of having access to proper respirology or immunology physicians. I was fortunate to be part of a study under Dr. Sin, so I'm one of the very few long COVID patients to have images to prove my lung scarring. Even with this, the narrative of the anxious female patient is dominating long COVID care. So she's added, I would love to see this research in the provincial post-COVID clinic.
Thank you, Patricia, and thank you for participating in this study. That's very appreciated. We're very grateful to all of our patients. Yeah, so we're really hoping to push this to clinicians. We are publishing in primarily, clinician-led journals such as the European Respiratory Journal and the Blue Journal, American Thoracic Society Journal. And again, we're speaking at conferences. Recently, American Thoracic Society conference just happened. A lot of my colleagues spoke there. I spoke at the immunology meeting in Chicago. So we are really trying to get the message out there because it is unfortunate that it tends to dominate, or long COVID tends to be prevalent in a group that is historically marginalized. Women, younger patients that, typically are told you're young, so you're healthy, high BMI, and again, ethnic minorities. So we're really hoping that we can help patients like you and hopefully make changes.
That's terrific. Yeah, all the best with those initiatives too. Another question, did you conduct the experiment without in vitro cell stimulation?
Yes, so there was no stimulation. The cells were so the PBMCs were collected directly from patients and then frozen down day of, and then they were run together in CyTOF. For the BAL, they were literally sent to the Biomedical Research Center, like fresh samples. We didn't even freeze them down. And yeah, we did not do any stimulation. That said, we are working on a co-culture model of the immune cells and the airway epithelial cells, and then we will possibly try some stimulation there to see, how interferon and other immune factors work.
Terrific. I'm just sorry to interrupt, but I'm posting the webinar survey and feedback questionnaire. If anybody would like to, please copy that link and send us any feedback. That would be terrific. So another comment and question from Nick. Thanks for the presentations. Neutrophils and YOT cell. Sorry, I can't quite read that.
Gamma delta, I think.
Okay, gamma delta, sorry, are highlighted in your study as mast cells. Can act as APCs, which activate the other cells, can activate neutrophils via cytokines, chemokines. Would it be useful to study mast cell activity? Sorry.
Yeah, I think it would definitely be useful. And the only thing is, in the BAL, we were not able to see the mast cell populations, and I was not able to profile them in the blood. So yeah, hopefully someone else can pick that up. Just unfortunately, due to the nature of our samples, mast cells are not something that we are studying. That said, we are currently hoping to recruit for an asthma cohort in the coming months. So definitely mast cells will be on our top priority. So we'll be looking at different ways we can try and capture those.
Terrific. Thank you. See Angela there. Any questions, Angela, from you or comments?
No, I just want to thank sort of Estefania and Elizabeth for great presentations. And I want to just make a couple of comments. So Estefania, maybe you and I can chat a little bit about sort of the RECLAIM samples. And so RECLAIM is an adaptive clinical trial in long COVID patients. And we collected samples as well as the CANCOV samples. And I know that a couple of people are here from that group working on something similar, but not exactly. So I think they can be complementary. And it's great to see these work. And so another thing that I want to mention to everyone is that we're trying to educate healthcare providers. And so so we want to give them a forum where they can speak up, ask questions, show their sort of ignorance per se. And so we're starting something called ECHO, Long COVID Web ECHO. And we're starting both English and French. So French is mainly in Quebec, but English is across the country. So we're working on that. I hope to start it in the fall. It's for healthcare professionals. So just want to let you know, and would love to have you both to come back to present, and so that this work can be shared with clinicians as well. Because for clinicians, they're so used to certain tests like MRI, CTs, chest X-rays, and often those are normal, and often even PFTs. So it's sort of often because of how they're taught, they concentrate on those tests. The tests that you did are more invasive, and in general, we don't sort of suggest to do it on everyone because of limitations of resources. So sharing those results will help understanding and also management of these issues as well. So would love to have both of you back for that series.
Thanks, Angela. And I see Nick, thank you, has put the ECHO Ontario link there for you to pass along to your friendly healthcare professionals and anyone online who's eligible there. Thank you. I think if there's no further questions, I'll just say a huge thank you to our two graduate students. Wish them luck in their thesis development and writing phases. But thank you for joining us today. And thanks to all of our participants for joining today. If you have just a couple of minutes, please do complete the feedback survey that we've posted in the chat there. Have a terrific afternoon, evening, morning, wherever you are. Thank you. Bye-bye.
Thank you, Susan.
Thank you, Susan and Angela. We will be happy to collaborate, happy to chat. Anything we can collaborate in, please contact us. Thank you.
Perfect. Thank you.
Bye.
Yeah.
Bye-bye now.